Since its inception, the human race has struggled with the preservation of food. This is especially true for things such as fruit and meats, which are highly susceptible to bacterial growth. The most common method, and the one our ancestors used, is to cook the meat over a heat source, killing all of the bacteria. But is this really the best way? This is the question that myself and 5 others asked, as we set out to determine the most effective method of preservation. We started with 2 types of meat, beef and tuna. The beef and tuna were both chopped up into pieces roughly 1 centimeter cubed, totaling 20 each. The cubes were prepared for preservation in the following manners. The first is heat. Next, we turned some of the cubes into jerky, using salt to dry the meat. Lastly, a ceviche was performed using lemon juice. After the meats were cooked, they were placed into agar dishes with 1 piece of beef and 1 tuna each. Agar dishes are designed to allow the bacteria on the food to grow. This will let us get a better understanding of how much bacteria was on the meat to begin with. Along with the 15 cubes of meat, we added in a control of both raw beef and tuna, as well as empty dishes. This gave us an idea of what we would be looking for in an optimal method of preservation. Now to get to the actual experiment. Here's how the dishes looked right after preparation. This is a control dish. And here is the control of raw meat. These 2 meats were ceviche'd. And lastly, the cooked trial. Unfortunately, no pictures of the original jerkied meats are available. The dishes were placed in an incubator for roughly 3 weeks. This is how they look after the fact. Here is a side-by-side comparison of the control dishes. There is some mold in the final control dish, but no bacteria. These are the raw dishes. The meat has since swelled and is infested with bacteria. These are the ceviche dishes. There is almost no bacteria present, as the remaining lemon acid helps to kill them off. Here are the cooked dishes. It is difficult to see, but the white specks are colonies of bacteria. On average, 10 different colonies can be found. This is the jerky after the experiment. The large white spots are salt crystallizations. Some bacterial colonies are present, though for the most part the salt levels are too high. Roughly 2 to 4 colonies can be found in the jerky. It would seem that without a doubt the best way to preserve food is by ceviche. The ceviche dishes have almost no bacteria, while the others have at least a few colonies. If you want to keep your food clean for the longest time, then out of these trials the ceviche is the most effective. It is easy to see how biology and chemistry are intertwined in this experiment. From a chemist's perspective, the experiment is good, because it examines the ways in which transformations of matter can help to preserve food, such as using salt to force osmosis on the bacteria. From a biologist's perspective, the experiment also examines the growth of bacteria over time, in several varying environments and with slightly altered food sources. It is somewhat difficult to truly separate every piece of the lab into either chemistry or biology, because they are incredibly similar. Advanced experiments often cover more than one branch of science. This experiment serves as a good demonstration of how the 2 build off of each other, utilizing chemistry to help get biological results and biology to get chemical results.